Hi, I'm Brett Zani. I'm formerly a postdoc in the laboratory of Dr. Elazar Edelman at MIT. And currently I'm a principal scientist and study director at a company that's affiliated with the Edelman Lab. We were actually trying to heal airway injury using various tissue engineering techniques when we started to notice these mysterious cell structures forming when we grew two distinct cell types together in the same culture dish. Those two cell types were human airway epithelial cells. Now these are the cells that line the inner surface of your airway. And the second type of cell was human endothelial cells. And these are the cell types that line the inner surface of your blood vessels. Over time, what happens is the two cell types actually segregate from one another. And to use a geography analogy, you have this ocean of single-layered endothelial cells covering the entire dish. And then sporadically placed throughout are these multi-layered epithelial islands. So this segregation was actually very interesting in and of itself. However, when you looked at higher magnifications, we actually started to see these very interesting long, thin structures connecting some of the epithelial islands to one another. And they actually hovered above the underlying endothelial cells. So in actuality, they were bridges. So that's why we call them epithelial bridges. So when we looked in the scientific literature, there was absolutely nothing on epithelial bridges whatsoever. So when this happens, it's kind of like a eureka moment. It's like, oh my God, did we actually find something completely new here? So from the serendipitous observation of these new structures, a whole completely new project was formed, with the broader goal being what is the function or functions of epithelial bridges. And as it turns out, epithelial bridges may facilitate cell migration by allowing one cell in an epithelial island to actually migrate from that epithelial island to the other connecting epithelial island. There's actually two reasons why I selected this image. The first reason is that it's one of the definitive images that shows that a cell is within a epithelial bridge, and that supports the notion that these conduits may be used for cell migration. The second reason is the aesthetics of it. First, you have these vibrant colors representing different aspects of life on a cellular scale. And then you have the 3D aspect of it, where the epithelial bridge and the top layers of the epithelial islands are in focus, while underneath, the underlying endothelial cells are out of focus. And that's because this is just a regular fluorescent microscopy image where you only have a single focal point. I think one of the fascinating things about these structures is that they're normal human, healthy cells. So if you took cells from me, you took cells from you put them on a culture dish together, they will form these structures. So the real question is, do these cells actually form in the body? We don't know yet, but it will be really fascinating to find out.